Hello everyone. In this video we will discuss demonstration of endosmosis, exosmosis and plasmolysis. Now let us start with aim of the first activity, demonstration of endosmosis and exosmosis. Demonstration of endosmosis and exosmosis. And the second step, apparatus. What is meant by apparatus? Apparatus is nothing but the materials required to perform this activity. So the materials required to perform this activity are: we need 2 petri dishes, raisins, water, salt solution. The third step in this activity is method. So in this method, first what should we do? We should take the 2 petri dishes. See, this is the petri dish A and this is the B. And in petri dish A, we should take plain water. In petri dish B, we have taken the salt solution. Now put dry raisins in petri dish A containing plain water and leave it for 30 minutes. After 30 minutes, what happens? The dry raisins placed in the plain water swells up. This is due to endosmosis. After 30 minutes, the dry raisins swells up due to endosmosis. We have already discussed endosmosis is the osmotic entry of water inside a cell or a system. Now we should place some swollen raisins in the salt solution. When we place these swollen raisins in the salt solution, after 15 to 30 minutes, the raisins will shrink. After 15 to 30 minutes, raisins shrinks. So this shrinking of raisins when they placed in salt solution is due to exosmosis. So what is meant by exosmosis? The osmotic exit of water from a cell or a system. Now let me write here method and observations of this activity. Put dry raisins in a petri dish having plain water. And observe after about 30 minutes. Observe after about 30 minutes. The next step in this activity is observation. The raisins swell up due to Endosmosis. The raisins placed in plain water swells up after 30 minutes due to endosmosis. And after this, what we have done, we have taken some swollen raisins and placed in concentrated salt or sugar solution. Some swollen raisins placed in concentrated salt solution. So after 15 to 30 minutes, what happens? The raisins placed in salt solution shrinks due to exosmosis. After 15 to 30 minutes, the raisins will shrink to its original form due to exosmosis. So this is one activity which explains endosmosis and exosmosis. Now let us do the another activity. Activity 2. Aim of this activity: demonstration of endosmosis and exosmosis. Apparatus or the materials required to perform this activity is 2 equal sized raw eggs, 2 beakers, dilute hydrochloric acid, water and salt solution. These are all the materials required to perform this activity. Now let us see the next step, method or procedure. Here first we should take 2 beakers. In one of the beaker we should take hypotonic water where solvent concentration is more and solute is less, and hypertonic corn syrup or salt solution or sugar solution should be taken in another beaker. After taking these two, we should keep them aside. Now place the equal sized raw eggs in dilute hydrochloric acid for 1 or 2 days until the egg shells get dissolved in dilute hydrochloric acid. When the raw eggs are placed in the hydrochloric acid, the egg shells which are made up of calcium carbonate get dissolved, and then later on we should take out the eggs from the HCl. We should wash them with water 2 to 3 times carefully, and here each egg is surrounded by a membrane, and we can call it as a plasma membrane or a cell membrane. It acts as the semi-permeable membrane. And after that we should place one egg in water. See, this is the egg without shell. So this is called as de-shelled egg. So we should place the two de-shelled eggs in the two beakers. So one egg should be placed in hypotonic water, another de-shelled egg should be placed in hypertonic solution, and leave it for some time. After half an hour, we can see that the egg placed in water swells up because of the osmotic entry of water inside the cell across the plasma membrane, whereas the egg placed in salt solution shrinks due to exosmosis. So here in this case, water leaves the cell into the external environment, that is into the hypertonic solution, because here solute is more than the solvent, and inside the egg amount of water is more than the solute. So this is why here exosmosis has taken place. Now let me write here. The first step is dip the two equal sized raw eggs in in dilute hydrochloric acid for 1 hour, and here we know that the egg shells are made up of calcium carbonate. They get dissolved when they are placed in dilute hydrochloric acid. Now we should wash the two eggs with water two to three times. And here each egg is now surrounded by semi-permeable membrane. Now we should place one of the de-shelled egg in water and the other in concentrated sugar or salt solution. So this is the procedure. Now let us see the observation. The egg placed in water swells up because of endosmosis. The egg placed in salt solution shrinks. The egg placed in salt solution shrinks due to exosmosis. After this, let us do the third activity. Here the aim of the activity is to prove the phenomenon of plasmolysis in the plant cells. Apparatus means materials required to perform this activity are rheoleaf, water, salt solution, glass slide, cover slip, blotting paper, microscope. Now let us see the method or procedure. The first step in this method is prepare peels from the purple colored side of rheo leaves. Prepare peels from the purple colored side of rheo leaves by twisting and tearing. Now let me show you the rheo leaves. Now look at this plant. These are the leaves of the rheo plant and its complete name is rheo discolor. Rheo discolor. See the name rheo discolor, but it is with different colors. You would have seen this type of plants in your school or in the parks in different places. Its leaves are a little swollen. So first what should we do? We should prepare a peels from the purple colored side of rheo leaves by twisting and tearing them. Here you can see the one side purple color and the other side is in green color. And the second step is boil one peel for killing its cells. Boil one peel for killing its cells. Keep the other peel fresh. Now mount the two peels on different glass slides. mount the two peels on different slides. So these are the two glass slides. From these two glass slides, first we should take a drop of water and we should place the peels of these two Rio leaves. We should uniformly spread the normal peel on one of the glass slide and the boiled peel on another slide. So this is A and this is B. On the glass slide A, we should place boiled peel of Rio leaf. On glass slide B, we should take fresh peel of Rio leaf. After this, the fifth step is, here the cells appear roughly similar in the two slides, having cytoplasm and colored sap. Cells appear roughly similar in the two slides, having cytoplasm and colored sap. And the next step is, replace the drop of water with a strong sugar or salt solution. Replace the drop of water with a strong sugar or salt solution. So this is hypertonic solution. So for this, what should we do? We should add a drop of solution from one edge of cover slip and water is removed with the help of blotting paper from the other edge. Here actually, after spreading the boiled peel on glass slide A and fresh peel of Rio on the glass slide B, we should cover them with cover slips. And so what should we do? We should replace the drop of water with a strong sugar or salt solution. And for this, what should we do? Drop of solution is placed on one edge of the cover slip and water is removed with the help of blotting paper. This is when glass slide is slightly tilted one side, water comes out from the other edge and that water can be wiped out by using the blotting paper. Now let me write here. For this, a drop of solution is placed a drop of solution is placed on one edge of cover slip and water is removed with the help of blotting paper. From the other edge. Repeat. Once or twice to ensure that the peel dips in strong solution observe after 5 minutes and observe after so we should add that strong sugar or salt solution to both the slides. For the slide A, on which we have taken the boiled peel, and another slide we have taken the fresh peel. So in these two peels we should add strong sugar or salt solution. So after doing this repeatedly for once or two times, we should wait for 5 minutes. So after 5 minutes, what can we observe? Let us take that under observation. When a plant cell or complete rheolith is placed in hypertonic solution, exosmosis occurs. When a plant cell or complete rheolith is placed in a hypertonic solution, exosmosis occurs. From the central vacuole of the plant cell, and we call it as plasmolysis. Now let me show you here the rheo peel which we have taken on the glass slide. When the normal water is added, the cells of rheolith will become turgid, and when hypertonic solution is added to the peel, so what is happening? There is a loss of water from the vacuole, so it results in shrinking of the cell, this by the exosmosis, and this process is called as plasmolysis. Here all the cytoplasmic contents along with the cell membrane is moving away from the cell wall, so this is called as plasmolysis. And this plasmolysis process can seen only in the fresh peel of rheolith. Whereas in case of a boiled leaf, there is no such type of plasmolysis occurs, because there the cells are already dead, so that it does not show any kind of exosmosis or endosmosis. But no plasmolysis occurs when rheolith first boiled in water for a few minutes and then placed in hypertonic solution. So this activity shows that only living cells possess semi-permeable membranes are able to exhibit osmosis. This is about demonstration of endosmosis and exosmosis with dry raisins. Next, demonstration of endosmosis and exosmosis with de-shelled eggs placed in hypotonic and hypertonic solutions, and plasmolysis in plant cells.